in healthy individuals. They are able to recognize and eliminate they are able to recognize and eliminate sites in order to respond to different microbes that cannot be destroyed by the innate immunity means. So, components of innate immunity include natural barriers and also include cellular and humoral branches similar to adaptive immunity branch, and innate immunity cells are represented by phagocytic cells and NK cells, natural killer cells, which are able to recognize in a non-specific manner common molecules expressed on microbial membrane, unlike the adaptive immune cells that recognize specific, very small microbial fragments. If you remember, I told you last Friday about them, epitopes belonging to antigens, and also include soluble factors, which are composed of microbicides, peptides, proteins belonging to complement systems, and also cytokines, which are secreted by cells belonging to innate immunity. Proteins belonging to complement system belong to humoral part of innate immunity, but also to the humoral part of adaptive immunity, because this system can be activated in two ways: in a non-specific manner, as in innate immunity, and in a specific manner, as in adaptive immunity, by antibodies which opsonize some bacteria, for example. Finally, after even if they have the two different ways of activation, they have a common path, which finally lead to destruction of pathogen by osmolarlysis. Okay, so let's talk about first line, which is represented by epithelial cells. The main entrance gates for all microbes are skin and mucoses belonging to gastrointestinal tract, respiratory tract, and urogenital tract. All of them have in common the following aspects. They are protected. All of these are protected by a continuous layer of each, represent the physical interface between environment and internal environment, and also represent the first layer of protection. They intervene, this layer of epithelial cells intervene from the first seconds after contact with microbes to block their entry into the body, and also they are able to remove themselves very quickly. So besides their role of mechanical barrier, they have selective permittivity for different substances, and also at the respiratory and the gastrointestinal level, they have motility, which also is involved in a protection mechanism. Epithelial barriers also provide the chemical means of protection at the entrance gates. These are based on the action of some natural antibodies, which can be found uh, uh, in some secretion. Also, at this level, we can find cationic antibacterial proteins, which include defensins and catalysidins. Both of these type of molecules inhibits the growth of microbes by direct lysis of their membrane. They have particular localization. For example, at respiratory tract, we can find beta defensin, and at the skin level, we can find alpha defensin and some catalysidins, some particular catalysidins. LL37, but it isn't important. It is important their role. Also, at this level, we can find enzymes which are able to destroy the pathogens. For example, there are enzymes which are able to destroy RNA, called RNases, and also DNases. And also at the derm level, we can find cryptidine, which are enzymes too. Another group of substances which are involved in protection in first line are colectins, which include surfactants, surfactant protein A and D, and also mannose binding lectin, which it's able to recognize mannose residues from some bacteria, triggering in this way complement system. Pentraxin is another substance which is able to protect the barriers. For example, C-reactive protein, which is able to inhibit microbe penetration by direct action, probably by damaging membrane through lysis. Also, this C-reactive protein can act as a chemoattractants, and also they facilitate ingestion and destruction of some microbes by phagocytes. Biological means are represented by commensal microbes. These can be found at the skin level, but also at mucosal level. The skin skin and intestine are colonized by more than 500 commensal bacteria species. Commensal microbes represent actually 95% of all cells of a human body. So we are made of microbes, after all. The role of this commensal species is to defend their territory, and in this manner defend our barriers. And also some of them have specific roles in some location. For example, in the intestine, the commensal bacteria participate to the digestion. These commensal bacteria can become pathogenic in certain circumstances. For example, in immunocompromised organisms, for example, in some skin at level, a skin level, in area considered sterile, destruction, and also this commensal bacteria can be destroyed by some medication. When we use antibiotics, some of these commensal bacteria are destroyed. So also at the levels of barriers, we can find lymphocytes, a particular type of lymphocytes, T lymphocytes, with which are disposed intraepithelial, and this particular type of lymphocytes have a particular structure of their receptors, TCR, T cell receptors. We'll see that if the common T lymphocytes have two alpha and beta chains in their receptors. These particular intraepithelial T lymphocytes have gamma and delta chains, and they are able to recognize a limited number of structures similar to pathogen recognition receptors. For example, they are able to recognize microbial lipids, and they will act as in similar manner as innate immune cells. Also, at intraepithelial level, we can find some particular subpopulation of B lymphocytes, which are called embryonal B lymphocytes. These B lymphocytes are able to recognize T-independent antigens. Also, they act in a similar manner with innate immune cells. So, what else? What else we have endothelial cells? In some conditions, cytokines and chemokines. These are peptides, very small peptides, which act as messengers. For example, cytokines transmit some messages related to the function of the nearby cells or the cells at the distance, and chemokines usually acts as chemoattractants for some cells in particular conditions when they are activated. So, in conclusion, epithelial cells belonging to first line of defense have the role to block the entry of microbes and destroy them through their specific means. Okay, so let's see what's happened. As I told you before, in normal condition, in physiological condition, postcapillary endothelial cells are impermeable to cells and plasma, but in some pathological condition, for example, when a pathogen succeeds to penetrate the epithelium layer and enter into the body, as a result of this penetration will be activated endothelial cells, and as a result of this activation they will be able to synthesize and secrete cytokines and chemokines. Cytokines released, the most important cytokines released are TNF-alpha and interleukin-1, which are known as pro-inflammatory cytokines because they are are able to change the morphology and adhesive properties and permeability to the endothelial cells itself, but also they are able to chemoattract some leukocytes in the from the capillary. As a result, we will have an inflammation, which is characterized by redness, pain, and loss of function, as you well know. This, for example, we have some pictures of inflammation, because inflammation can be seen in any tissue. For example, in here it's a normal lung image and pulmonary edema as a result of inflammation. Here I want just to name the sequences of leukocyte migration, because all of these steps and all of the steps of inflammation are discussed in the physiopathology lecture in extenso. But I want to remember you the sequences of leukocyte migration. As I told you before, as a result of their activation of endothelial cells, they express a different type of cellular adhesion molecule, which will be able to establish some bonds with leukocytes on the one hand, and first step is to decrease distance blood circulatory, and next is to activate integrins and to establish a stable adhesion. And after that, the next step is to activate migration through the endothelium, and in this way will contribute to the defense of the to destroy the pathogen which already entered through the barrier. Let's talk about innate immunity cells. So as you already know, the leukocytes sorry about that. Leukocytes can be divided in granulocytes and agranulocytes, and depending on their cytoplasmic content. So the granulocytes have the following general characteristics. They have a multi-lobate nuclei, and also their cytoplasm contain numerous granules, which these granules can be stained with different dyes. Basic proteins from granulocytes can be stained with acidophilic, eosinophilic dyes. So we will talk about eosinophils. Amines stains with basic dyes. So we will discuss in that situation about basophils. And there are some cells which can be stained with neutral color dyes, and we discuss about neutrophils. They have different functions. All of these granulocytes have different functions, but neutrophils are most known phagocytes, non-specific phagocytes. and they are the most numerous and are effective in killing bacteria. The eosinophiles intervene in allergic reactions as well as basophils and mast cells due to their content of vasoreactive amines. The most known is histamine. Also, you should keep in mind that the physiological role of eosinophils are anti-helminthic defense. In agranulocytes, they are characterized by a large nucleus, and they don't have a cytoplasmic. They function as lymphocytes. We can find two types of lymphocytes. Large lymphocytes, which are natural killer cells, and small lymphocytes, which can be T lymphocytes or B lymphocytes. After their activation, these small lymphocytes can drive different type of immune response. Humoral immune response have the effective cells, plasma cells, which derived from B lymphocytes, and these cells are able to synthesize and secrete antibodies. And on the other hand, the effector cells of cellular mediated immune response are cytotoxic T lymphocytes along with helper 1 cells. Okay, so also derived from a myelocytic line, we have antigen-presenting cells, mostly dendritic cells. They have multilobate nuclei containing the cytoplasm granules that are colored with neutral color. They are non-specific phagocytes, being the most numerous, and they act their main role is to present antigens to the specific lymphocytes. Also, with originate in myeloid line, we have monocytes, which will be transformed in macrophages after their passing into the tissues. And the macrophages also, they are able to act as antigen presenting cells. Just a few just a few details about these cells. Monocytes can be found in around 1 million monocytes in 1 milliliter of blood. As I told you before, they are able to migrate into the tissues and differentiate to macrophages when they will have a different name related to their resident tissue. For example, if they are located in liver, they are called Kupffer cells. In lung, they are called alveolar macrophages. Also, they can be found as microglial cells in central nervous systems, and in bone, they are called osteoclasts. Their role, the main role, is to phagocytose microorganisms, but also, as I told you before, they can act as in blood of action of these cells. They have first stage is to identify the pathogen, and for this they will work with some receptors which are expressed on their membrane. These receptors are able to recognize some ligands. As I told you before, the ligands can be any molecule which have a conformational compatibility with a receptor. And the receptors which can be found on innate immune cells are able to recognize only some molecular patterns of pathogens, or pathogen-associated molecular patterns, which have, from a structural point of view, these are common structures for all microorganisms and cannot be found in mammalian cells. This is the most important characteristics of these pathogen-associated molecular patterns. From chemical point of view, these PAMs can be nucleic acids, for example, double-stranded RNA structures, which can be found in viruses. Also, non-methylated CpG DNA sequences, which can be found mostly on bacteria organisms. Also, there are some proteins specific to bacteria, for example, azote-formyl-methionine structure. Also, as you know from microbiology, there are complex structures made of lipids and oligosaccharides synthesized by some microbes. Gram-negative bacteria, for example, synthesize lipopolysaccharides, but also many residues are a combination of lipids and carbohydrates. From functional point of view, these pathogen-associated molecular patterns are essential for survival of microbes. This is very important because these microbes cannot give up, modify the structures in order to avoid recognition by the immune cells. So this sentence is in reality is not entirely true, because we already know that some viruses are able to change their molecules, surface molecules, and the very to have a high variation from a season to season of their molecules. For example, gripal viruses, rhinoviruses actually, which change their structure, which can be recognized every season, and for this reason we have to vaccinate every season against these viruses. Okay, so but actually these molecules belonging these PAMs, most of them belong to some important functional structures of microbes. Let's talk about some types of pathogen recognition receptors. On the other hand, we have these receptors which are able to recognize these PAMs. There are mainly four types of pathogen recognition receptors. The most largest one is toll-like receptors, which can be localized on membrane on extracellular membrane, but also in nuclear membrane of some macrophages, but also can be seen on dendritic cells, on B lymphocytes, endothelial cells, and other cells. For examples, we have toll-like receptors 1, 4, 5, and 6, which are able to recognize lipopolysaccharides and also peptidoglycans. The second type of pathogen recognition receptors are C-type lectin-like receptors, which can be found on plasmatic membrane of macrophages and polymorphonuclear cells, meaning phagocytes. And those receptors, C-type lectin-like receptors, are able to recognize carbohydrates, mannose or fructose residues, terminal residues, through mannose receptors. Also, there is a type of C-type lectin-like receptor, which are called tectines, which are able to recognize a specific structure belonging to fungi, which are glycans. The third type of pathogen recognition receptors are scavengers receptors, which can be found on a plasmatic membrane of macrophages and polymorphonuclears. And for example, is CD36 receptors, which is able to recognize deacylglycerate structure expressed on some microbes. And the fourth type is azote-formyl-methyl-leucine phenyl receptor. As you can see here in its name are named the aminoacids residues, the sequence of aminoacids, which is recognized by this receptor. And these receptors are expressed also on phagocytic cells and are able to recognize this particular sequence of aminoacids, which can be seen in some pathogen-associated molecular patterns. The toll-like cell receptors, toll-like receptors distribution. Toll-like receptors are expressed predominantly on cell types that are likely to be first encounter of antigen, as we already seen. On phagocytic cells are most of them expressed, and there are a lot of receptors were described 18 receptors till now, but not all of them are well characterized. Phagocytic cells such as macrophages, neutrophils, and dendritic cells exhibit a broadest repertoire of these receptors and express the highest level of toll-like receptors. However, toll-like receptors expression is not restricted to these cell types. Also, toll-like receptor expression patterns and levels in most cell and tissue types are still being defined. It appears that the majority of cells in the body expressed at least a subset of toll-like receptors. Toll-like receptors 3, there are conflicting reports regarding their expression, in particular leukocyte population. Some of these reports suggest that toll-like receptor is only expressed on dendritic cells, while others find that toll-like receptors are expressed on NK cells or even T cells. And also these receptors can be upregulated, an upregulated expression on epithelial and endothelial cells. It has been shown that neutrophils express all toll-like receptors except toll-like receptor 3. Toll-like receptor 3, it's able to recognize double-stranded RNA, which is characteristic to viral infection. Also, neutrophils aren't usually associated with viral infection. They are associated with certain respiratory viral infection. Therefore, it is unclear by which mechanism neutrophils would directly recognize virally infected host cells. Okay, we should keep in mind that toll-like receptor 4, for example, is expressed on most phagocytic cells and also in antigen-presenting cells, but they are not expressed on lymphocytes. Toll-like receptor 8 are expressed on monocytes and in low levels on NK cells and even T cells, and toll-like receptor 7 can be found in B cells and plasmacytoid precursor of dendritic cells. These are the most important toll-like receptor cell distribution. Their distribution related to the cellular membrane. They are 9 receptors, 9 toll-like receptors, which are very well characterized from the point of view of their membrane distribution. It was found that 4 of them, toll-like receptor 3, 7, able to recognize structures common to viruses, such as double-stranded RNA or single-stranded RNA, but also some of them, toll-like receptor 9, for example, it's able to recognize CpG structural motifs, methylated CpG structural motifs, which are characteristic to bacterial DNA. Some of these receptors are expressed on the extracellular membrane, and if you're looking here, you'll see that they recognize some structure characteristics of some exogenic antigens. For example, toll-like receptor 1 and 2 recognize lipopolysaccharides, but LPS, characteristic to gram-negative bacteria, are recognized by toll-like receptor 4. It's important to keep in mind that toll-like receptor number 2 recognize peptidoglycans, and for example, toll-like receptor 5 recognize flagellin. If you look in here, you will see that in their structure, all these toll-like receptors are dimers. Yes, all of them. And besides pathogen-associated molecular patterns, they are able to recognize also some molecules which are called damage-associated molecular patterns. For example, HSP, which is heat shock protein. There are some chaperone proteins which are expressed when a tissue cell suffers some stress. For example, the stress after a viral infection, and also HMGB1. High mobility group box 1 molecule, which is also associated with viral infection stress. Here is a table which summarizes receptors and their ligands, characteristic ligands. And if you look in here, you can see that their ability to bind, for example, viral molecules is related to their location. Okay, please note that there are some toll-like receptors able to recognize fungi. Toll-like receptor 6, which is able to recognize a zymosan, a molecule expressed in fungi. Also, toll-like receptor 9, it's better to keep in mind because this is specific. They can recognize these molecules characteristic to bacteria, but also to some viruses and to malaria agents. Okay, so how they works? How this receptor works? Toll-like receptor function. As I told you before, they are dimers. All of them are dimers. As a result of combining conformational binding of the ligands, pathogen-associated molecular patterns, or damage-associated molecular patterns, they will change their conformation, and this change can be seen either in their intracellular part. This intracellular part is in very high contact with some adapter proteins, for example, MID88 or TRIF proteins, myeloid differentiation factor 88, or toll immune receptor domain containing adapter inducing factor, and also can be and also as a result of this attaching with these adapter proteins, these proteins will suffer also conformational modification, and this will lead to activation of a cascade of phosphorylases, which finally will allow to be activated some transcriptional factors, for example, interferon response factors or NF-kappa B. As a result of activation of transcriptional factors, will be increase the rate of transcription of some molecules. for neurons. As a result of their secretion, the cell will be able to fight against the viral infection, for example. Other factors which can be secreted as a result of activation of these transcription factors are some cytokines or some cellular adhesion molecules such as TNF alpha or interleukin 1, which promote inflammation, but also they are able to synthesize and to release cellular adhesion molecules, which will increase the number of leukocytes able to fight with the aggressor. So, functional response of pathogen recognition receptors, no matter what type of pathogen recognition receptors, is, as I told you before, after recognition, which induce some conformational changes in the receptor. These changes will be it will include the intracellular part, because in order to be a receptor, a molecule has to be able to bound, to recognize, to identify molecule, but also should be able to transduce, activating a signal through different pathways, which will lead finally to cellular response. In this situation, functional response of pathogen recognition receptors include increase expression of cellular adhesion molecules, which will lead to migration into the tissues of some leukocytes. Also will increase the production of cytokines, but also will increase the production of active weapons. For example, if we are talking about phagocytes, will increase the production of reactive species of oxygen or nitrogen, which finally will lead to killing the microbes, but also will increase the ability to engulf the microbe, to phagocyte the microbe. After the phagocytosis, you know that next step is to have fusion between a phagosome and a lysosome, which lysosome, as you well know, contain enzymes which are able to destroy the pathogen, and also this will lead to destroy the membrane and also to destroy the pathogen. So, okay. The function of activated macrophages. We take macrophages as a representative of phagocytes, and they contribute not only to the killing of microbes, but also by their secretion of cytokines. As I told you, they are able to secrete TNF alpha, which is a pro-inflammatory cytokine, will increase inflammation, and through the secretion of interleukin 12 will be able to contribute to the enhance of adaptive immunity. Also, macrophages are able to secrete some growth factors and enzymes which contribute to tissue remodelating, and because of their ability to process the pathogen, to process the antigen, macrophages are able to um, um, synthesize also major histocompatibility complex and to present the non-self molecule epitopes along with MHC molecules in order to stimulate a specific lymphocytes when we need to trigger an adaptive immune response. So, here are depicted the interaction between phagocytes and other innate immune components. For example, here is a macrophage which phagocytose a pathogen. As a result of this phagocytosis, this macrophage will be activated, on the one hand to destroy the pathogen, on the other hand to secrete some cytokines. Macrophage is able to secrete interferon, for example, but interferon is able to activate, to increase activation of NK cells, which, as you know, NK cells are able to recognize an infected tissue cell, and this leads to destruction of infected cells. But after destruction of tisular infected cells, we need to repair the tissue, and as a result of activation of these cells, will be released also some growth factors, angiogenetic factors, and proteinases able to repair and to remodulate the infected tissue. Natural killer cells are large lymphocytes, non-T, non-B. We call natural killer cells as large lymphocytes because they have between 9 and 15 microns compared to 6 to 9 microns of small lymphocytes, T or B lymphocytes. They don't express classical antigen receptors. This is very interesting. But they recognize and kill abnormal cells such as infected, viral infected cells, but also tumoral cells, because they are able to recognize damage-associated molecular patterns, and also they are able to recognize self molecules expressed on our tissues. They destroy target cell either directly through releasing some enzymes, granzymes and perforins, which lead to membrane lysis, but also they are able to destroy target cell indirectly by releasing some cytokines such as TNF alpha and interferons, which induce apoptosis in target cell. Other cells involved in innate immunity cells are eosinophils, basophils, and mast cells. For eosinophils, we know their role of defending against parasites, but for basophil and mast cells, we don't know their physiological role. We think that they are involved in protection of mucoses, but we know for sure that these cells contribute to allergic reactions. Here is an image of electronic microscopy of schistosome larva, which is attacked by a lot of eosinophils and is covered with IgE antibodies. So next, I present you on very short the interactions between phagocytes and other innate immune components, such as complement. For example, as you know, the complement system can be activated on 3 different pathways. 2 of them are in a non-specific manner because they are able to recognize structures expressed on microbes, such as lipopolysaccharides or mannose residue, and the complement system also can be activated by recognition of antibodies, specific antibodies, which already opsonized the pathogen. Either of which way is activated, the complement will pass through the final steps, late steps of activation, which finally will lead to lysis of microbe by polymerization of a complement and constitute of membrane attack complex in the membrane of the pathogen. But on this pathway of activation of complement are released a lot of components with important role in the innate immunity. Components with role which act as chemoattractants, which act as opsonins, and also there are some components released which act as vasodilators. Timing of innate immunity after infection. So as you can see, I told you before that innate immunity have the advantage that acts very quickly after the contact with the pathogen. We need just seconds to block the entrance of the pathogens through the barriers, but if the pathogen succeeds to penetrate the barrier, we need just few minutes in order to activate epithelial cells and complement systems, and we need minutes today to secrete cytokines in order to signal that an enemy is in the body, and we need just few hours to activate neutrophils to recognize the pathogen and to destroy them. But the disadvantage of neutrophils is that they have very short life. We need hours to days to activate macrophages. And also we need hours to days to activate natural killer cells. Cytokines, I told you a lot about the cytokines which are released as a result of activation of these innate immune cells. The most important cytokines released by these innate immune cells are tumor necrosis factors, but these are released not only by macrophages but also by T cells, and they will act. The most important action is on endothelial cells, which lead to activation of these cells and also activation of neutrophils. But the TNF have also systemic influence because they induce fever and also induce hepatic level synthesis and secretion of some other pro-inflammatory cytokines such as interleukin-6, but also reactants of acute phase of inflammation will be synthesized and released. Interleukin-1 is the other pro-inflammatory cytokines which can be synthesized by many sources and also increase the activation of endothelial cells and increase the fever and synthesis of the acute reactive phase activating factors. Interleukin 12 is very alpha because they contribute to cellular mediated immune response. As I told you, interleukin-6 is the third pro-inflammatory cytokines with a major contribution in increasing the inflammation. And interleukin 15 and 18, which are secreted mostly by macrophages, activate other cells. So we can discuss later the details of mechanism of immune invasion. And now I want to present on shortly the cooperation between innate immunity and adaptive immunity, because starting from next week, we will discuss all the details of an adaptive immune response. So as you already seen, after the entrance of the pathogen, the first layers of defense, which include barriers, but also cells and humoral factors belonging to innate immunity, will fight with the pathogen, but this the cells of innate immunity include macrophages, which also are able to process pathogen and to present epitopes to the specific lymphocytes. And also dendritic cells belonging to innate immunity are able to present epitopes to the specific lymphocytes, which is the first step in order to trigger an adaptive immune response. So that's all for today. I hope that you already you understood. If you have some questions, please tell me. No?
No, I think it's okay, doctor.
Hello?
No.
I didn't understand.
No, it was clear, doctor.
Okay. I didn't see your... did you sign in the chat? Okay, I find it. I didn't see in chat anything. You didn't sign in chat in order to
We
have
started writing our names at the beginning of the lesson. So most of us already wrote our names down.
Okay, I don't know why I did. Oh, okay, so I find it. Okay. But some of you entered at 11 and 18. I don't think that's... and some of you entered at 11:25. Good, very good, like this. So you understand that it's impossible to have an attendance in this situation, yes? Okay, so we'll meet again next Friday, and we will discuss about antigen presenting cells. Okay.
I have a question. Will we be receiving any other information? Like, can we read from something else?
Sorry?
Like, will we be receiving any other documents to study from besides the PowerPoints?
I told you from the beginning, you have a bibliography. So you will receive this material, and also you have a bibliography. I recommended you a bath and sleeping coat, so I think that you don't need the supplementary material, for example, for this one. It's all in here. It's a table. What
Okay.
else do you need? You have to read it. So, oh, okay. So, another question? Other question, please?